Much, Jane. That was wonderful. Our next speaker is Sofia Lozano from UT San Antonio, and the title of their talk is Novel Peptide Drug Demonstrates Anti-Coccidiodes Activity.
Can everyone hear me? Good morning. My name is Sofia Lozano. I'm a master's student in Dr. Hung's lab down at the University of Texas in San Antonio, and I'm excited to be here today talking to y'all about a novel peptide drug that's been demonstrating some anti-coccidioides activities. After all of the wonderful presentations yesterday, I'm sure cocci needs no introduction, so I'm going to take a little bit of time to talk to you about the different classes of antifungal drugs. Specifically, I want to bring your attention to these two that are here in the middle. Polines, such as amphotericin B, bind to a gestrel and disrupt the cell membrane, whereas azoles, such as fluconazole, target the ergastral biosynthesis pathway. We are starting to see an emergence of fluconazole resistance in a growing number of clinical isolates. So due to this limited therapies, the need for a novel class of anti-coccidiodal drugs is unmet and urgent. We are fortunate enough to be in collaboration with Dr. Michael Lorenz from the University of Texas Health Science Center in Houston, who brought to our attention some really interesting data. Their lab was able to characterize that Enterococcus facialis produces a bacteriocin known as ENTV, which has been proven in the past to affect the virulence of Candida albicans. As seen by the figures here on the right, ENTV increases the survival in their C. elegans model in both azole-susceptible and azole-resistant strains. Additionally, a shortened 12-amino acid version of this original amphipathic peptide has been shown to also increase survival in their C. elegans worm model. While it's really interesting that this works in Candida, we wanted to know, could this potentially be effective against coccidioides? Very interestingly, ENTV not only is effective in Candida albicans, but as well as Candida aureus and Cryptococcus neoformans. As seen by the figures here on the left, there is still an increase in the C. elegans survival in both Candida aureus and Cryptococcus neoformans. What was most interesting about this is that the treatment with ENTV leads to differing outcomes within these fungal species. In Candida albicans, it inhibits adhesion and hyphal morphogenesis, whereas in Cryptococcus, it works by binding to the extracellular vesicles. Given how evolutionarily distant these two fungi are, with Candida being an ascomycete and Cryptococcus being a basidiomycete, there is the potential that the unknown target of ENTV could be widely conserved among several different fungi. For this reason, we have other collaborators who are testing its effects in Aspergillus and in Mucor. Well, it's great that this works in the C. elegans model. What was most interesting, though, was seeing how this works in a mouse model. The Lorenz lab was able to challenge mice with Candida albicans, wait 3 days to induce infection, and on day 3, dose the water supply for these mice with 200 micromolar of a modified version of ENTV known as P4L here. The treatment lasted for 2 days, and on day 5, qPCR was done to see what the fungal burden was. As seen here in the figure on the right, this modified version of ENTV lowered the fungal burden in multiple of their clinical isolates for Candida albicans. As I mentioned, this is a modified version of ENTV, so to talk a little bit more about how it was modified, 2 different stereoisomers were derived from an even shortened 10-mer version of this ENTV. You have your L stereoisomer, which are more commonly found in nature and are more susceptible to protease degradation, as was seen in the previous slide, using P4L. P4D was created in order to prevent this proteolytic activity in being the D stereoisomer. In fact, it helped to enhance peptide stability, giving the peptide a half-life of more than 24 hours in human sera. As seen here in the figure on the bottom right, there's a drastic increase in the amount of the peptide being found in human sera. So, as a real quick recap, in Candida albicans, it's batting 1,000. In Candida aureus and in Cryptococcus, it is showing really promising work. So our goal became to test to see if this could be done in coccidioides. So our next question became, is there an equivalent mini model to the C. elegans that could be used to initially test coccidioides? Luckily, we have the Galeria melanella model recently established by the Hung lab, which worked as a great initial test. See, this model is cost-effective, easy to maintain, and reduces the amount of mice, making it great for these preliminary studies. But what was most important and what was most interesting is this coccidioides can complete its entire life cycle within this organism. As seen by the figure here on the right, you can see various different morphologies of coccidioides, including active endosporulation. So what we did is we took a very small needle and we're able to inject these Galeria worms with 4.5 times 5 to the times 10 to the 5 CFU of CP C735 spores. 2 hours post-infection, we treated with a total of 10 microliters of P4D, and we watched their survival for the next 7 days. And what we saw is that here in this middle figure, there is an increase in survival with our Galeria that were treated with P4D and infected with CPC735. In fact, not only is there an increase in survival against our PBS negative control, there is also an increase in survival between our P4D and our amphotericin B control. We did also test all 3 of these treatments in Galeria that were not infected with CPC735, just to ensure that there's not going to be any adverse reaction with the Galeria. Additionally, this peptide P4D lowered the fungal burden in comparison to PBS, not quite as low as amphotericin B, but still statistically significantly lowered from our negative control. So we wanted to start to kind of unravel how and why this works. So what we decided to do was to test to see the metabolic effects of P4D using an XTT assay. After culturing spheroidal initials, we seeded a drug plate with various different concentrations of P4D, set it to incubate for 24 hours, and after those 24 hours, we added XTT. See, what XTT does is in the same way that mitochondrial dehydrogenase reduces NAD plus to NADH during oxidative phosphorylation, it can reduce XTT to formazine. XTT typically has a pale yellow or clear color, whereas formazine has this bright orange color, working as a colorimetric indicator for cell viability. Essentially, if the cell can metabolize XTT to formazine, we know that the metabolic pathways are not being inhibited too terribly bad. So after 24 hours, we read the absorbance. What we saw is that in our amphotericin control, you see this nice dip in metabolic activity, indicating that it is inhibiting metabolism in the spheroidal initials. However, in our P4D, it stays pretty consistent regardless of the concentration of P4D, indicating that P4D does not inhibit spheroidal metabolism. Given that in the previous studies done in Canada and in Crypta, it is working as an antivirulent agent. This is kind of what we were more or less expecting to see with this P4D, that it's not kind of inhibiting that metabolism. We also wanted to see if it was altering the visible morphology or size. So we do have pictures here of RS in our highest concentration of P4D that was tested, which was at 40 micromolar after 24 hours with treatment, compared to its corresponding negative control, which was 4% DMSO. And as you can see, the size of the spherules, the shape, the overall conversion did not alter after being treated with P4D in comparison to its negative control. So in conclusion, P4D does not show any inhibitory activity via XTT assay in coccidioides, indicating that the drug is not fungicidal. Additionally, it does not visibly alter morphology or size. In the Galeria model, it has been shown not to be toxic at a dose of 0.01 micromolar, but it does increase survival and it lowers fungal burden. My future directions would be to start to do this in an in vivo study using a murine model, additionally to redo the Galeria experiment with other clinical isolates, as well as a wider variety of concentrations in order to optimize that dosage. Additionally, I want to be able to identify where P4D is interacting with coccidioides, so testing it via fluorescent microscopy to get a better idea as to where it's binding, which has previously been done in both Candida albicans and Cryptococcus gattii. Finally, to test P4D's metabolic effects in other morphologies, such as arthrochonidia and segmented spherules. Simply because it does not work in one does not mean that we can rule out all of them. I wanted to take this time to acknowledge and thank all of the members of the Hung Lab. I would not be here without them, as well as all our collaborators in the Michael Lorenz Lab. And finally, I want to thank all of you for your time and attention, and I'll be happy to take any questions.
Much shorter. Hi, great talk. Are you able to identify any functional groups of P4D that may be displaying this anti-cocci behavior?
We have not yet, but it's definitely something that we're looking into soon.
Okay, thank you.
Hi. What stage is the cocci at when you inject the drug at 2 hours, and why was that time point chosen as opposed to later on?
So we injected them with arthrochonidia with spores. We waited 2 hours post-infection so that if you essentially treat the Galeria back to back, it can cause, like the injury and the trauma to the worm can cause them to die a little bit earlier. So we did wait a couple of hours to allow them to heal before treating them with P4D. So the initial injection was with Arthrochonidia spores, and the second injection was with the treatment.
So when you're treating them, it's arthrochonidia, not in the spheral stage yet.
No, yes, but because cocci can undergo its entire life cycle, we do start with arthrochonidia, and we watch kind of how the infection progresses over the next 7 days, where it does have the ability to kind of complete its entire life cycle.
Could you treat it later when it's maybe a spheral in the Galeria model?
It's definitely something that we could try. As I mentioned, I do want to redo the Galeria experiment, so maybe adding another group would be beneficial. Thank you for the suggestion.
I had a question kind of piggybacking off of that. You saw that it still did the endospores and everything and made spherals. Did they rupture? Do you know? Did you check?
Not for this specific model, but because we know that's something that's been seen and well documented in the past, we are kind of working with the assumption that it is kind of constantly completing its life cycle within the Galeria.
Oh, I meant, did the peptide inhibit rupturing or anything? Did you?
We have not seen that. That's one of the reasons why I want to do this in kind of fluorescent microscopy at different time points to be able to see kind of where that peptide is binding. Thank you. Thank you all again.